Hello everyone, my name is Sunyub Kim, and I'm excited to explore AlphaFold with you today. It's the groundbreaking advancements in protein structure predictions. This topic today is closely related to the bioinformatics concepts that we have been studying for the semester. To begin with, let's discuss what AlphaFold is. It's developed by DeepMind, is a part of Alphabet, and a developer of Gemini. It made headlines in 2018 during the 13th CASP, Critical Assessments of Structure Prediction. This global competition challenges researchers to predict protein structures from amino acid sequences. AlphaFold 2 achieved remarkable success, outperforming all competitors and showcasing the incredible potential of AI in scientific research. And recently, Demis Hassabis and John Jumper, the creators of AlphaFold, received the Nobel Prize in Chemistry, highlighting the transformative impact of AI on traditional scientific fields. The development of the model is going on real time. The team just disclosed the next proprietary model, AlphaFold 3, both paper and the source code, just a few weeks ago. It is pushing the boundaries of biotech industry continuously. Now let's dive into the central challenge that AlphaFold addresses. It's a protein folding problem. This task involves predicting how a linear sequence of amino acids will fold into specific three-dimensional structure. Each amino acid has unique properties that influences its interactions, ultimately determining the protein's final conformations. Proteins are composed of 20 different amino acids, each playing a distinct role in the folding processes. Some amino acids will going to attract each other while others repel, guiding the overall folding mechanism. Due to the complexity of this problem, before the advent of prediction models, most conventional methods, such as X-ray crystallography and spiral electron microscopy, taking months or even years to determine the structure of proteins, and costing unsustainable costs to the researchers as well. So how does this AlphaFold solve this problem? It employs two primary components in its predictions: multiple sequence alignments, which is MSA, and pair representations. AlphaFold enhances protein structure predictions by utilizing evolutionary data, particularly through multiple sequence alignments. MSA aligns homologous protein sequences from different species, allowing for the identification of conserved residues, which are the amino acids that remains unchanged over time. For instance, human, horse, and fish versions of hemoglobin demonstrate significant sequence divergence. However, yet their 3D structures remain largely conserved. This structural conservation indicates the importance of certain residues for protein stability and its function. The conservation of specific positions suggests their critical roles in folding, catalytic activity, or ligand interactions. Additionally, the coevolution of amino acid residues can indicate interactions, further informing structure predictions as well. By analyzing these evolutionary patterns, AlphaFold effectively predicts protein folding, achieving results that rival the experimental methods. Now let's discuss pair representations. This component encodes the spatial relationships between amino acids, capturing not just the distance and angles between them, but also potential interactions between pairs of residues in three-dimensional spaces. AlphaFold determines this representation through iterative updates during the prediction process. And initially, it uses placeholder values based on the amino acid sequence. Then it refines these values based on observed interactions as the model learns from the data. Additionally, a key innovation in AlphaFold is its attention mechanism. This feature enables the model to focus on the most significant amino acids within a protein sequence, weighting their importance based on their potential impact on the overall structure. Having understood how AlphaFold works, let's move on to the advancements introduced in AlphaFold 3. There are three key advancements I would like to highlight today. First, AlphaFold 3 exhibits an impressive ability to generalize across various molecular types while simplifying its underlying model. This version significantly expands its capabilities, moving beyond individual proteins to include interactions with multiple proteins, DNA, RNA, and ligands, which are all potential drug candidates. By focusing on atomic positions and interactions rather than rigid geometric relationships, AlphaFold 3 achieved versatility that makes it applicable to a broader range of scientific inquiries. Next, in AlphaFold 3, the role of MSA has been adjusted. It now serves as a supplementary tool to inform the pair representations, which focus on how pairs of atoms interact. This strategic shift allows AlphaFold 3 to analyze molecular interactions more effectively and efficiently, handling larger sequences than before. Researchers recognize that reducing iterations in the MSA component would still yield crucial information, making the process much more faster without sacrificing the accuracy. Another major advancement in AlphaFold 3 is the introduction of the diffusion model, a model that is akin to fine and AI image generators. Essentially, it mimics how particles spread out over time, reflecting natural molecular interactions. The process begins with known atomic position of the protein. Similar to dropping food coloring into the water, the model adds noise to these positions. It allows atoms to move unpredictably, much like the color dispersing. As the model explores various arrangements, it evaluates which configurations are more stable, gradually refining the atomic positions. This iterative process leads to a more accurate predictions of the protein structure. Rather than strictly adhering to rigid geometric constraints, this approach enables the algorithm to deduce optimal positions through a more flexible probabilistic method. As we look to the future, the potential of molecular modeling is very promising, with models becoming increasingly generalized and capable of handling diverse inputs and outputs. We may soon enable predictions about molecular behaviors and interactions in real world applications. So, as a conclusion, the evolution of AlphaFold to version 3 represents a monumental leap in our understanding of protein structures. By embracing simplicity and generalization, we stand at the threshold of a new era in molecular modeling. And such advancements could lead to significant breakthroughs in biotechnology, further expanding the frontiers of molecular science. And this is all about it. Thank you for your attention today, and I hope this presentation helps you find your next step in bioinformatics. Thank you.